Thank you very much for attending. I will hand it over to Dhananjay, who will start off the session.
Right, thanks, Chandrakant. And wherever you're viewing this from in the world, hello and welcome to the opening talk for this season of the Bangalore Developmental Biology Club. I'm Dhananjay. I'm a postdoc at NCBS here in Bangalore, and we'll go through the seminar together. We're very fortunate to have with us biologists with careers full of scientific discovery, and you for participating in today's proceedings. We'll first hear from Sonia, a member of faculty at TIGS, about the aims of BDBC, Bangalore Developmental Biology Club. Professors Vijay Raghavan, Vidyanand, and Scott Gilbert will have a short discussion about the creation of the textbook of developmental biology, the future of the field, and some very pertinent questions that we, I think, should all be thinking about. And then we'll then come to the seminar that we're all here for by Professor Gilbert, followed by a question and answer session. So any questions that come to mind, please put them down in the Q&A box and we'll get to them at the end. The BDBC truly hopes that we can get you to participate actively in this and subsequent activities. And so, Sonia, could you please tell us about the plans of BDBC and how we plan to go forth?
Yes, of course. Let me share my screen. Okay, can someone confirm that they can see my screen?
Yes, we can.
Yep.
Okay, and you can hear me. Okay, great. Okay, so as Dhananjay said, this is the first of the seminar series of the Bangalore Developmental Biology Club. And I have to say that we've been calling ourselves this, but in reality, turns out that a lot of the people who've registered for this are actually not in Bangalore, from a lot of places in the world. So, okay, my slide. Okay, oops, sorry. Yeah, okay, yeah. So we'll just say that we call ourselves this because it's a group of developmental biologists in Bangalore who run this, but it's really open for anyone. And particularly all of our virtual events, if you're interested, do please join us, and you can follow us on Twitter to know what the activities are. So what is it that we have planned? So the idea of this forum is to really bring the community together and to strengthen the community. What do we mean by this? So the first thing is that you get to invite speakers from anywhere in the world and get to listen to them on a monthly basis. So we will have this every month. The other activity is that you get to be a part of the community, means that you can get feedback on your work, on your as you progress through it. So we'll have forums where people can present their work and get feedback on their work as they're developing their projects. Then we plan to have journal clubs to keep track of new developments in the fields. Initially, it'll just be journal clubs, but we're planning to make this into a pre-review journal club where we are reviewing preprints and posting reviews onto the pre-review website. And the idea here is that we develop the ability to give critical, constructive feedback on papers and learn to read papers with that in mind. Okay, then finally, very importantly, to be able to share reagents within the community. This is really important in a place over here. Then as you're developing your grants and writing your papers, before you send them out for review, you can ask your community for feedback on these as you're writing them. And then finally, to collaborate. So as you meet and you talk to each other, you realize that you have shared interests, you have complementary expertise that you can come together and work on a problem together. So you find collaborative community. And then, of course, just to have fun. Okay, so how are we planning to do all of this? The idea is that you have, even in Bangalore, you have a whole bunch of institutes that have different labs that work on different questions. And the idea is to bring all of these different labs together on a regular basis to be able to do all of these activities that we spoke of. So we will be meeting every Friday at 4 o'clock for an hour and a half, no more. And and we will cycle through these activities. The work talks will happen on the first Friday of the month. So we'll circulate between the labs, where each lab gets to present, a member from a particular lab gets to present their work. The second Friday of every month, that is today, like today, we'll have a seminar series, and each lab can get to choose who they want to invite, and we'll facilitate that. And then the third Friday of the month will be the journal club, the pre-review journal club. And then the fourth Friday will be just hang out and have fun. I guess that'll have to wait for some time, but yes, that's the idea. And then of course there are some months with 5 Fridays. And so on those months, we will, 3 times a year, we will have a symposium type of setup, which allows people to explore the talk formats that you'll commonly encounter in any meeting, international meeting that you go to. So the lightning talks, the platform talks, or the plenary talks. And the idea of the symposium, it'll be theme-based, but the idea is to basically bring together people and give them experience of being able to give those formats of talks. So upcoming, the next few Fridays, the 16th, that is next Friday, we're going to have an organizational meeting. We'll post a link to that meeting in the chat box over here. So do join us if you want to be part, and I urge you to be part of this community. Then, unfortunately, the next Friday is the 4th Friday, so we cannot meet right now, but we'll be off planning the rest of the Fridays. Then July has a 5th Friday. So we're going to have the first symposium of this season, and you'll soon hear what the theme of this particular symposium is. And do send in abstracts. We'll publicize that very soon. And then the next Friday will be an off Friday because we don't work 2 Fridays in a row when we have a mega symposium like that. And then the next Friday is actually a seminar series. We have a very exciting speaker lined up. We're finalizing the detail of time across different time zones, and we'll post that soon on the various forums that you get your information from. So stay tuned. Okay, so while all of that is happening, we have a Slack community to keep in touch with each other. So we'll also post a link to the Slack so that you can join there. When you do, please introduce yourself with a little bio and update your profile with a picture so that we recognize each other in a pandemic virtual world. And over on the left, you'll see the various channels related to the different events. Do go ahead and join those. So we'll keep the community talking to each other even between all of the times that we meet. And then get involved. Please do get involved. So you can do that by either DMing us on our Twitter handle or writing to any one of these 4 people who are currently running it. But of course, we want all of your to be involved. So do please write to us and get involved. Okay, with that, I will stop and hand it back to Dhananjay.
Right, thank you, Sonia. I really think that everybody on this talk should be getting involved in this club, because I think it's going to be a very rewarding experience. So in this next part, we'd like to talk a little about writing the textbook and the future of the field. For this discussion, we have with us Professor Nanjundaya, who investigates the development and social behavior of slime mold Dictyostelium. He is based at the moment at ICTS in Bangalore. Welcome, Dr. Vidyanan.
Thank you.
Joining us is distinguished Professor Vijay Raghavan, a Drosophila developmental and neurobiologist from NCBS in Bangalore. Don't think he needs much of an introduction. He is also the principal scientific advisor to the government of India. Hello and welcome, Vijay.
Thank you.
Our speaker for today is distinguished Professor Scott Gilbert from the Swarthmore College in Pennsylvania in the US. Apart from his, fairly well-known work, the textbook on developmental biology, he's worked extensively in the fields of evolution and development. honestly, the list of honors and awards of our guests is long. It's really long and readily searchable. So we're just lucky to have them with us. And I think we'll move straight to their scholarship and with a brief discussion before the talk. And from here, Vijay, could you please take over?
Thank you very much, Dhananjay, Sonia, and others. It's really fantastic that you've started this Bangalore Developmental Biology Club. And what a great start to have Gilbert as your first speaker. Scott, of course, is known to all of us. And, when I started teaching developmental biology at NCBS, I ordered his book and Walpert's book. And unfortunately, Walpert's book arrived earlier. And so I used it for my first course, and I was caught in the spandrels of San Marco trap. And having used it the first year, I had to use it the next year and so on. But I always had an introduction in the beginning about the history of developmental biology, and all of that pretty much the modern history was from Scott's work. So I'd like to start off by, asking Scott to referring him to a paper he wrote in 1988. It's called The Cellular Politics, Ernst Everest Just, Richard Goldschmidt, and an attempt to reconcile embryology and genetics. And this was, the three main pillars of modern biology: developmental biology, that's embryology, genetics, and evolution. And getting them all together has transformed biology today. But Scott's work in 1988 looked at, how Just and Goldschmidt helped in the transformation. Just, a Black American, after coming back from Europe to America for a visit, said that in 6 months in Europe, he saw more collegiality than he saw in a lifetime in America. And Goldschmidt, coming to America from Europe, said in Berkeley that in a few years in America, he saw more collegiality than he saw in Europe. So politics and biology were intertwined. And I hand it over to Scott to give his comments on that.
Well, thank you. Thank you very much, everybody. Sonia, Chandrakant, Vidya, thank you for inviting me, and best wishes on the Bangalore Society for Developmental Biology. This is wonderful. Thanks to Vidya, I've had some wonderful experiences in Bangalore, and I'm just sorry I could not physically be there today, but really best wishes on your new society. Yeah, the turn of the last century, the years around 1900 were absolutely fascinating for the separation of evolution, development, and genetics into three different sciences, because they had been kind of linked together as one science. The motor for evolution was development. Things developed, and if they developed differently, they could become new forms of life. And so development, especially in the theories of someone like Ernst Haeckel, were critical for evolution. And the way that development proceeded was through genetics. Now, the early geneticists were all embryologists. This is something which I found fascinating as kind of, student of the history of the field, that people like Morgan, people like Bateson, they were originally embryologists. And they saw genetics originally as a subset of embryology. The question that genetics asked was, which compartment of the fertilized egg, the nucleus or the cytoplasm, controlled development? Where are because what you inherit is a developmental process. So which part of that cell, which part of the fertilized egg, the nucleus or the cytoplasm, had the determinants? And people like E.B. Wilson said, it's the nucleus. And people like Boveri said, it's the nucleus. And their evidence was largely based on sex determination. They said, look, in the nucleus, there is an X chromosome. If you have two X chromosomes, your development is in the female direction. If you have one X chromosome, it is in the male direction. So there's something in the nucleus that controls development. And people like Morgan, Thomas Hunt Morgan, was actually in favor of the cytoplasm. Before 1911, Morgan was the largest American proponent for the cytoplasm. And he said, yes, but there's something that tells the polar body to have an X chromosome or not. You know, that it's the polar body, it's something in the cytoplasm, which is telling the cells how to divide during meiosis. And they really argued about this until Morgan got the evidence that on the X chromosome were the genes for wing shape and eye color. And then the easiest way of explaining this was that these were determinants physically on the X chromosome. And his lab then ran with that. So Morgan, in a large way, was responsible for the separation of embryology and genetics. It became a field with its own language, its own organisms, and so forth. It became separated from the field of embryology. At the same time, people like Wilhelm Roux were separating embryology from the field of evolution. Roux wanted embryology to become a discipline like physiology. It wasn't going to just be evidence for evolution. It was going to have its own questions, the question of what is the physical mechanism by which the fertilized egg becomes the adult. And we didn't need to be tied to evolution. And so you had at this time embryology saying we're not tied to evolution and genetics saying we're not tied to embryology. So you have the separation of three disciplines from what used to be a single discipline with one investigator not having to choose what they were doing. It was how they interpreted their data more than it was whether they were an embryologist, a geneticist, an evolutionist. What of course was happening is that at the same time, biology was being professionalized. It was a field that you could actually make money in. You could actually have a job in biology. And so the question was, who was going to be hired? Who was going to be hired by a university? A geneticist, an evolutionary biologist, an embryologist. And so you had the separation of these people professionally. They wanted to be known as one thing and not the other. So there was a lot of kind of, institutional politics being played, but there was this real honest notion of we have new questions. The question of what is it in the nucleus? What are these chromosomes doing to control development? And how do you get evolution from these genes? And how does the fertilized egg become an adult? You have the separation of what was the field of heredity into these three aspects. And this was new. Then in the 1920s, people tried to resynthesize them. And that's when you get people like E.E. Just, Richard Goldschmidt, Conrad Waddington, and Salome Gluckson-Schönheimer, all trying different strategies to integrate this. Goldschmidt looking at the genes, E.E. Just looking at the cytoplasm, especially the cell surface. And I think it was Waddington and Salome Gluckson-Walsh who really combined Spamann's embryology with Morgan's genetics and made a field which we would now call developmental genetics. Developmental genetics was the term that Salome Gluckson-Walsh-Schönheimer coined in 1938 to explain what she was doing. She was not doing embryology, which was taking an egg and making some sort of experiment on it and seeing what happens. No, she was looking at what the genes did in their experiments and seeing what is the earliest stage where you got a defect in development. She called it developmental genetics. Waddington called it physiological genetics, but they were the two who really integrated these fields together, and they did it both by looking at the notion of embryonic induction. They found genes responsible for embryonic induction. And so they really fused the fields for the first incarnation of developmental genetics. It's intellectually a fascinating field to look into. Most of these people were friends. And that was another thing. The geneticists who became embryologists, they were still friends with the embryologists. It wasn't an antagonistic set of interactions. It was a very friendly set of interactions. And that, I think, was very important for the entire field.
Thank you, Scott. We could just go on this topic,
but
easily. Vidya, give his points in, and then let you go on for your Holobiont talk. Vidya.
Well, I'm not sure what to take up. Scott and I have been discussing something which may be of wider interest here, which on the face of it has nothing to do with biology, which is something that's been written by Dipesh Chakrabarty, who is a historian, sociologist, anthropologist, everything. And he has this very interesting way of suggesting that you could bring together different time scales, the time scale of human cultural change and the time scale of geological change or climatic change, and how the two are fusing together in effect. Yeah. And Scott and I have been talking about it. Maybe you could share your thoughts on that, Scott.
Yeah. Oh, well, Chakrabarty mentioned that the current era, the Anthropocene, which may have started in 1492, it may have started with the steamship, but this technological age where humans can control nature has broken down the traditional boundaries between human history and natural history. Now, you cannot think of human history without thinking of nature. You cannot think of natural history without thinking of humans. And I think this has some very profound ways of changing how we usually think about natural history and about our cultural history. And I was just reading a paper last night, which I thought was fascinating in this respect. It was a paper by Bruno Latour, and he is saying that global climate change, whether it's manifest in tsunamis, in hurricanes, in the rise of the oceans, in the changing of the acidity of the oceans, it all has to do with one thing, and that's thermodynamics. That the changes in why you get all these hurricanes, why are you getting these storms, it has to do basically with the world trying to find a new equilibrium in a very physical thermodynamic sense. The world is looking for a new equilibrium. We humans have taken it out of its normal equilibrium. Buffers are going away. How is nature finding a new equilibrium? I think that's a fascinating way of looking at modern history, because what we're seeing is the migration of peoples due to climate change. The Syrian war was due largely to times of drought. We're seeing refugees trying to come into the United States, the southern border, because of the heat in Guatemala and southern Mexico. We're seeing movements of people. We're seeing movements of wind. We're seeing movements of water. And it's due basically to the Earth trying to find a new equilibrium. I think that when we look at, the conquest of the Western world by Europeans. That was done primarily by microbes. You know, the Pizarro's army did not number many people. Cortez's army did not number many people, but they wiped out 90% of the indigenous population. Well, they were bringing over viruses and bacteria. That was what wiped out the population, much more so than the guns and steel that the Europeans were bringing. And here we have a situation where the same microbes were parasites on the native populations, but they were symbionts and mutualists for the colonists. Whether or not the bacteria was good or bad depended on who you were. Same thing with, you know, I did a project recently on oysters. And the oysters are being saved because they can be useful for humans to save places like Chesapeake Bay. When I was in Baltimore, the Chesapeake Bay Society had a big, you know, bumper sticker, "Save the oysters." Now the bumper sticker is "Save Chesapeake Bay." How are they saving Chesapeake Bay? They're using the oysters as partners. The oyster can filter the water. So it isn't so much save the oyster, it's save the bay. And the oyster is our friend. Our oyster is our co-companion in saving the bay. So I think we're seeing more and more of these partnerships with nature to do things. It's a different way of looking at nature. It isn't seeing the oyster as a resource, it's seeing the oyster as a partner. Our histories are coming together in a major way. So that's, I think what Dipesh Chakrabarty was saying is really profound. And I think it's a way of looking at the world right now that's a very useful and satisfying way of looking at it because it brings together so much data. It's like a good scientific model. Yeah.
Thank you.
Oh, you're welcome. Again, we can go on and talk about this for hours at least today.
Thank you, Scott. Dhananjay, do we have time or should we go on to Scott's talk?
I think we should move to the talk.
Okay.
We have a question and answer session later, so
Good.
we can continue the discussion.
Let's have Scott.
Oh, that would be fun.
Vidya's last point was a nice segue into his talk itself. So it'll be wonderful to get Scott going. Thank you.
Okay.
So are you going to reintroduce Scott or does he just get started?
I could just get started.
We know him, so yeah.
Okay, I will go to my screen share and see if this works.
Over to you, Scott.
Okay, see if. Okay. Okay, can you, let's see, can you see my screen?
Yes, we can.
The elephant on it. Okay, well, thank you again for inviting me. I'm going to be talking about the evolution of holobionts, and I'll explain that in a moment. But I want to look at how symbionts during development can initiate new trajectories of evolution. And the setting for the talk will be how we can evolve by acquiring new genomes. This was a hypothesis. This was an idea that was brought about by Lynn Margulis and her son, Dorian Sagan, back in the 1990s. And basically, my talk will be structured on the following: that animals are holobionts. Each organism is both an individual organism, but also a biome, a consortium of interacting species. I will also say that animals develop as holobionts, that we do not develop from the fertilized egg with its nuclear genome. That is part of our development, but we also use symbionts to control other parts of our development. So even our development is not monogenomic. It does not come from a single genome. It comes from dozens of genomes. Third, I want to talk about how animals can evolve by acquiring new genomes. I'll first talk a little bit about microevolution. I'll talk about microevolution in a bug and a beetle and how symbionts actually give it new evolutionary properties. And then I want to talk about macroevolution. And I want to talk specifically about the evolution of the cow, the ruminant. Cows are ruminants. And I want to figure out, how did the rumen evolve? How did that most important part of the cow's stomach evolve? So, let's see, where try to go here. Okay, so let's start off by talking about holobionts. Animals are consortia, assortments of numerous species, and are therefore both organisms and biomes. Each one of us is a collection of numerous ecosystems. We're biomes. And I have here some of my favorite representative holobionts. There's the cow, and the cow is eating grass, turning it into milk. But we know that the cow, as a cow with a cow genome, cannot digest grass. No mammal can digest grass. We have no enzyme encoded in our genome for digesting cellulose, pectins, or hemicelluloses. Okay. What digests the grass are the bacteria, archaea, and fungi in the cow's rumen, in that part of its stomach. So the cow needs the bacteria in order to survive. Corals. Coral needs algae in order to survive. That's why they grow at high depths in the water column. That coral needs an algae in order to undergo photosynthesis and give the carbon resources to the coral. If you bleach the coral, if you have coral without algae, they die. And coral bleaching is one of the major things going on in the Anthropocene. We have lost about half of our coral in the past 20 years due to global warming. Global warming upsets the nutritional balance in the coral, and the symbionts leave. Mixotricha paradoxica, Mastotermes here, Mastotermes darwinensis, is a termite. It is a pest. It eats wood. But there's nothing in the termite genome that allows it to eat wood. What eats the wood is this protist in its digestive system called Mixotricha paradoxica. And Mixotricha is here, but Mixotricha is itself not an organism. Mixotricha is a combination of 5 separate organisms into one. What looks like cilia here on the outer borders of a protist, well, there is a protist there, but it doesn't have cilia on its outer border. These are spirochetes. These are bacteria that allow it to move. So the holobiont describes the host and its microbial communities. And the microbes can be constant, they can be temporary, they can be transmitted vertically through the egg, they can be transmitted horizontally through the environment. I'll get to those later. The host and the microbiome function together. And this is critical. They function as a unit. We develop as holobionts. Our immune system is not a host system against bacteria and viruses. It is a holobiont system against particular viruses and bacteria. But it is not just the host, it is the holobiont. Our physiology is regulated in part by the bacteria. And so the fitness of the host, I will say, depends on the mutual interactions between the host and the microbe. We're not talking only about the big guy. We're talking about the hundreds, the thousands of symbionts. So when I say here that the holobiont is who we are, that we are a biome, a collective of ecosystems, we're saying that over 50% of the cells in our body are microbes. And these microbes are not randomly located. They have specific locations. Each pore on our skin is an ecosystem. There are about 160 species of bacteria in each of us. If you look at the whole human population, there are about 1,000 different species of microbes that we routinely carry on us. And again, the different parts of our bodies have different microbes which have formed communities. Okay, how are these microbes transmitted? There are 4 ways. One is vertical transmission through the ovary. Okay, if I were to show the slide on the left to anybody in my developmental biology course, I would hope that they would say, oh, this is a Drosophila ovarium, and you have this red cell over here. That's going to be the oocyte, and these blue cells, they're transporting material. They're the nurse cells transporting materials into the oocyte, and they'd be absolutely correct. But what these red dots are, these things that are being transferred from the nurse cells to the oocyte, these red things are not bicoid mRNA, they're not ribosomes, they're not nanos mRNA, they are Wolbachia bacteria. The oocyte is collecting bacteria which will be used in its immune system. So one is this vertical transmission through the egg. Another way is horizontal maternal transmission through the birth canal. And here we see a giraffe being born. And as it's being born, its amnion has ruptured. It is being colonized by the microbes coming from the birth canal of the mother. It is getting bacteria as it is being born. Now, we humans have about 22,000 different genes that we get from our parents. We get about 8 million different genes from the bacteria. Bacteria differ in their rates, but we can largely say that a bacterium has about a 70% chance of being transmitted from mother to offspring. Another way of going from mother to offspring is horizontal transmission through coprophagy, through poop, through excrement. And here we have the koala bear, and the koala mixes its milk with its feces and smears it over the face of its baby. And so the baby gets what's in the feces of the mother, almost 100% transmission from mother to infant. Another way, the 4th way, is strict horizontal transmission. And this is where the environment gives the symbionts to the offspring. The classic example here is the Euprymna squid. This is a Hawaiian squid, and it's born without a light organ. But as soon as it's born, its ventral surface gets colonized by Vibrio fischeri bacteria. And these Vibrio fischeri bacteria interact with the skin of the belly and create the light organ of the squid. Now, as Lynn Margulis liked to pick up to talk about this, saying that this is not just a gentle process, because all the other bacteria that tried to colonize the squid are killed. They are oxidized, they are tanned, they are sloughed off, so that only functional Vibrio fischeri bacteria are able to get into the organism. But we have this interaction going on between the squid and the bacteria to make a squid organ that the bacteria house themselves in, and the bacteria produce the light to the light organ. So this is this notion that we are holobionts and that we have a physiological way of transmitting the holobionts, of transmitting the bacteria. The bacteria can be seen as non-nuclear inheritance. They are something that gets inherited from one generation to the other. And there are physiological mechanisms for doing this. Now, physiologically, you have in our body, or we have in our bodies, multiple organisms acting for a common good. Each of us can be considered a team. And this is a very common metaphor used in holobiont bacteria. Each of us are teams. And here we have this team, a Planococcus Planococcus insect, a mealybug, and its pathway for making the amino acid phenylalanine starts in its symbiont, Tremblya bacteria. But the Tremblya bacteria, a rather large bacteria living inside Planococcus cells, has a bacteria living inside it. The Morinella bacteria lives inside the Tremblya bacteria, which lives inside the skin of Planococcus. Okay, to make phenylalanine, the reactions start in the symbiont, then they pass into the symbiont's symbiont, where more metabolic interactions take place. They go back into the symbiont, and finally, the last stage of phenylalanine production is done by the insect's genome. So you have this kind of nested Russian doll system of symbiosis. We have that in our bodies too. The microbes that are in the digestive tract, that are in our gut, are important for the function of the neuroendocrine system, the digestive system, the circulatory system, the immune system, the kidneys, and the bones. The microbes are in part responsible for the maturation and maintenance of our digestive, neuroendocrine, circulatory, and immune systems. We act as a team. About 70% of the human resting body temperature is probably the result of gut microbe metabolism, and over 95% of the serotonin in our bodies comes from bacteria. That serotonin, by the way, as we'll see, is necessary for the peristalsis of our gut. So bacteria that we get in our bodies is mostly a gift from our mother. During the last trimester of pregnancy, the hormones change the population of microbes in the female reproductive tract, such that if you were to give first trimester microbes to germ-free mice, they stay very normal. But if you add third trimester pregnancy, late pregnancy from humans into germ-free mice, the mice get fatter, they get insulin desensitization, they basically get metabolic syndrome, which actually is probably helping the woman when she's pregnant. After the baby is born, the mother gives the baby milk. And the milk has two sets of nutrients in it. One set of nutrients is obviously for the baby. Here's the nutrients. Good, enjoy. The second set of nutrients in mother's milk is a set of oligosaccharides, of complex sugars that no mammal can digest. They're not for the baby, they are for the baby's bacteria, especially bifidobacteria. They're for the bacteria that the mother just gave the baby. And you want these good bacteria because bifidobacteria and others are the bacteria which set up the conditions for other bacteria to colonize and get into the gut, things that we'll talk about in a moment. Okay, so the microbes are critical for our health. One physiological example, the microbes regulate the peristalsis of food through our guts. The gut microbes make short-chain fatty acids, propionic acid, butyric acid, acetic acid. They make short-chain fatty acids. The gut epithelium absorbs these short-chain fatty acids from the gut, and it tells the gut epithelium to make serotonin, transform tryptophan into serotonin. Serotonin gets made, and serotonin is a hormone that gets released and will take those immature neurons, those neural crest cells that are in the gut, and turn them into the mature neurons, which can cause peristalsis to occur. So, as the title said, gut microbes regulate serotonin release, reception, and neural maturation in the myenteric plexus of mice. So the bacteria are used, they are needed by the bodies, by the mammalian bodies. This is important because development in mammals is incomplete at birth. If you take a newborn mouse and you look at its gut, at its gut villi, you see that there's a very poor circulation in the gut villi. The green represents capillaries. But if you add the guts, when you add the bacteria, you get a great capillary network, which can take the food from the villi of the gut into the rest of the body. This is what you get without microbes. This is what you get with microbes. The microbes are responsible for gene expression in the endoderm cells that causes capillary formation. What gene is being expressed? It's the gene for angiogenin 4. So if you look at this graph here, this is mRNA abundance, where 1.0 is germ-free. So this is without bacteria, 1.0. This is mRNA for angiogenin 4. Germ-free mice set it for 1.0. Conventionally raised mice have 10-fold that amount of mRNA. That they have all the gut bacteria. If you add gut bacteria back, you get about this 10-fold increase in what is the germ-free level. In other words, not having bacteria is like having a mutant that gets rid of 90% of the efficiency. 90% of the mRNA is coming because of the bacteria. And mainly that mRNA is being induced by Bacteroides theta iota micron, a very common species in the gut. It's basically giving you what the conventionally raised condition gives. It induces the expression of angiogenin 4. So animals do not exist as independent entities. Development involves numerous interacting species. The word for this is symbiosis, becoming with. We become with the other. We are not independent autonomous entities. We have co-development. It's not just physiological. It's not just interactions between consenting adults and their microbes. No, we form ourselves by interacting with others. We are never independent. Now, those bacteria, they're helping us. The Bacteroides induces angiogenin 4 mRNA in the Paneth cells of the intestines. That's what we just saw. The angiogenin 4 protein is seen in the Paneth secretory cells, and it gets secreted, and it tells the mesoderm surrounding the capillaries make capillaries. Surrounding the villi, it tells them to make capillaries. But, and here's something really beautiful, angiogenin 4 has another use. Angiogenin 4 is toxic to Listeria and Candida, two potential competitors of Bacteroides. So Bacteroides is not only helping us, it's helping itself. It's telling our gut cells, hey, you know how to make angiogenin 4, do me a favor. I'm gonna do you a favor, you do me a favor. You make this angiogenin 4, you kill Listeria and Candida. I don't want them around, they're competitors. So it helps itself as it helps us. The symbionts help construct our immune system. We usually see our immune system as separating us from the outside world. And we usually see it as a host against a potentially hostile environment. The new studies have shown that the immune system actually is a symbiotic, is a function of the holobiont. The bacteria help construct the immune system. The immune system is both bacterial and eukaryotic. For instance, germ-free animals have impaired development of the Peyer's patches in their intestines. They have a reduced number of naive T helper cells, reduced number of T regulatory cells, and a reduced tolerance. Peterson said, these results underscore the adaptive immune system's critical role in establishing a sustainable host microbial relationship. Well, This isn't the function of the immune system that I learned. I learned the immune system kills bacteria, and Peyer's patches is our first internal line of defense against hostile bacteria. And here I'm finding is that Peyer's patches helps the bacteria find a place to live. Obata says this may involve the creation of an optimal symbiotic environment on the interior of Peyer's patches. Whoa, this immune functioning part of the intestine, these Peyer's patches immune cells, they are helping the symbionts. Yes, later they are critically important for getting rid of microbes, but these are the microbes that the host and the symbionts agreed should be gotten rid of. Donaldson said gut microbiota utilized immunoglobulin A for mucosal colonization. I had learned that immunoglobulin A bound to bacteria and viruses like polioviruses to get it out of the body and neutralized. Now I'm learning that the gut microbes actually use immunoglobulin A to stay in the body. This is a new function of the immune system. It's not only to kill bacteria and viruses, it's to protect certain bacteria and viruses. Now, in 2013, a major paper was published by Margaret McFall-Yai and her 26 colleagues here. It was published in PNAS and it calls Animals in a Bacterial World, a New Imperative for the Life Sciences. Now that word imperative is hardly ever used in science. An imperative, this is something that must be done. You cannot do it otherwise. And PNAS allowed this, a new imperative. One of the things it says in this wonderful review, and it's still accurate today, but one of the things it said in 2013, it says, whereas developmentally important symbiosis have been documented throughout the post-embryonic, larval, and juvenile stages of vertebrate and arthropod life cycles, the roles of symbiotic microbes during normal embryonic development are just beginning. In other words, the animal might be born, the human, the mouse might be born in an aseptic environment, but it gets its microbes immediately upon birth, and those microbes complete the task. What hasn't been seen in 2013 is the microbes acting before birth. We now know that they do. Paper here, 2020, maternal gut microbiota in pregnancy influences the offspring metabolic phenotype in mice. Basically, what's happening here is that dietary fiber is utilized by the gut microbes of the mother, and short-chain fatty acids, propionic acid, butyric acid, acetic acid, are made. They go into the maternal circulation, they cross the placenta, they get into the mother, and here they bind to the G protein coupled receptors for short-chain fatty acids, GPR41 and GPR43, and they induce neural cell development. They make the sympathetic nerve projections to the heart. They increase the pancreatic beta cell development, and this protects them against metabolic disease in adulthood. The insulin-making properties of the beta pancreatic cell are needed, and they get regulated through the maternal microbes and the maternal diet. So even before birth, the mother's microbes are important. And this was shown by using a mother, having a pregnant mother with microbes, without microbes, having them be foster mothered by a normal mouse. And what you find is that the pups that were born from the germ-free mice get fatter almost from birth, that their metabolic rates are very different. The pups of the germ-free mothers were germ-free during pregnancy get fatter. And it was the diet, it was shown to be the short-chain fatty acids, which were responsible for insulin levels. So that if you look at insulin levels here, you see that the high fiber diet, those animals with high fiber diets, and the microbes were able to make more insulin. Okay. Propionic acid stimulates pancreatic beta cell development and insulin production, and it's the GPR43 fatty acid receptor which is needed for this induction. And so insulin is made by the mice that have the bacteria, not by the germ-free mice or the mouse with the mutation in the receptor for the fatty acids. Okay, so mother's bacteria influence the offspring's development in utero. We saw this again, 2020. The maternal microbiome modulates fetal neural development in mice. The maternal microbiome promotes neural axon growth, and that's shown here. This is your mouse with bacteria and SPF, just as we know the bacteria that are in this guy. And if you look at the neurites here, you could see that here's your normal case with the bacteria. Here's where you add antibiotics, and here are the neurons with the bacteria being abrogated by the antibiotics. And here's germ-free mice, that the neurites are growing. And these are neurites that are involved in hearing. So the metabolites from maternal gut microbes are able to induce the development of these neurite connections. In the absence, they do not make as many connections. And that can be shown behaviorally. These animals which had the high fiber diets and had the microbes, they get startled by a loud sound. However, that startle reflex is not done by the animals which did not have the metabolites produced by the microbes and the fiber. And it seems like Clostridium bacteria are the ones that are critical for this. So we now have a situation where microbes are critically important for the development of adult behaviors. This is also seen, by the way, in social behaviors. Germ-free mice have autism-like behavioral symptoms. So germ-free mice would rather be in an empty chamber than be with other mice. So here we see there's the time spent, the differences between time spent alone or with other mice. Normal mice like to be generally with other mice. Germ-free mice do not, but if you add bacteria back to the germ-free mice, they go back to liking other mice. If you look at compulsive behaviors in mice, you see very little in the bacteria, in the mice with the bacteria. You see a lot more obsessive behaviors in the mice without the bacteria. If you add the bacteria back, you lessen their compulsive behaviors. This is being looked at now. There's a pilot study that's come out in humans as well. So we now know that we are holobionts, that we develop as holobionts. So if evolution is brought about by inherited changes in development, that's what Evo-Devo is about. And if development is caused in part by symbiotic microbes, that's what I was just talking about, then changes in developmental functions of the symbionts may cause changes in evolution. And there is evidence for this. This is something that Lynn Margulis was talking about when she said that life did not take over the globe by competition, but by networking. And at the highest level taxa, they have evolved by the acquisitions of symbionts that have become hereditary. And as she said once in an interview, I don't consider my ideas controversial. I consider them right. Other people may consider them controversial. But evolution by genome acquisition. And we know that different symbionts can give different abilities. And here we have studies by Dunbar and Moran's lab on the P aphid. The P aphid is parthenogenic. There's no males. It just makes, by changing its meiosis, it makes more and more of these female aphids. Now, Bucknera is an obligate symbiont of these aphids. However, there are allelic differences in the Buchnera genome. Some Buchnera symbionts can give thermotolerance because they encode a heat shock protein. Other Buchnera symbionts do not encode a functional heat shock protein. They do not make it. So in the range where the P. aphid lives, you can select different populations of P. aphids by heat. And this depends on the allele of their symbiont, such that if you look at fecundity over most of the range, most of the temperature range, the fecundity is about the same with the mutant, with the non-producing allele being slightly favored. However, if you have high temperatures, the symbiont that cannot make the chaperonin protein, that cannot give the thermotolerance, is selected against, whereas there's no selection against those holobionts that have a symbiont making the chaperonin protein. So here is selection that can be done not on the allele of the P. aphid, but on the allele of the P. aphid's symbiont. We have another fascinating example in the bean bug Riptortus. Now, the bean bug Riptortus has nymphs that live underground, and they acquire Burkholderia bacteria at each generation. The second instar larva eats the Burkholderia bacteria, incorporates it into tissues that are in its digestive tract, and there it resides for the rest of its life. Now, farmers wanted to kill this bean bug because it was eating the soy plants. So they sprayed insecticide. They sprayed a certain insecticide on the field. What they didn't know is the Burkholderia bacteria could use the insecticide as food. So in addition to getting rid of the bugs, they were giving the Burkholderia bacteria that could digest this pesticide. It gave that a huge advantage, so that the few bugs that survived were now eating Burkholderia bacteria, which could digest the pesticides. And the bugs that incorporated the pesticiding Burkholderia became resistant to the pesticides. So Riptortus, the bug, is selected as a holobiont. Even if the Burkholderia can exist independently, they've now helped the bug to survive. Okay. The red turpentine beetle. This is an innocuous beetle that I could find if I go out of my apartment and walk into the forest in the Pacific Northwest. It's a minor pest. It is a beetle that makes its home in dead trees. And it helps actually digest the dead tree and make soil out of the dead tree. It's not a big problem. However, it got shipped over to China, probably in furniture or railroad ties. It got shipped over to China, and in China, it met a different fungus, because what digests the holes in the wood is not the genome, not the enzymes from the red turpentine beetle. They're enzymes produced by the fungus that's a symbiont of the red turpentine beetle. And the red turpentine beetle in China found a new fungus, and this fungus makes it deadly. It makes it kill living trees. Over 10 million trees have been destroyed in China by this beetle. It became a pest by changing its symbionts. Its evolution has been altered. So I want to end by talking about macroevolution and developmental symbioses that may be facilitated by changing the symbionts. Especially, I want to talk about the evolution of plant eaters, of herbivores. Now, herbivory comes in late into evolution. Most people kind of think that there are herbs and there are the herbivores that eat them, and there's the carnivores that eat herbivores. And that's the way it is now in the food chain. But originally, there were filter feeders, there were detritivores, and there were carnivores. It's easy to eat animal flesh. It's difficult to get energy from cellulose and hemicellulose and pectins of the plant. And the widely held view that the overall pattern of trophic evolution has been from plant grazers to carnivores has its origins in such pedagogical tools as Elton's ecological pyramid and Odum's food chain. But this is an ahistorical comparative morphology. Herbivory comes after carnivory. Herbivory is late. Herbivory, it comes late. Plant eating is the derived condition, not meat eating. Within the ecdysozoa, the plesiomorphic state is primarily carnivory. That's what we begin with. In vertebrates, predation was often the ancestral condition from which herbivory evolved. The clear implications of our findings is that the trophic level of macrophagous primary consumer, a herbivore, is a relatively late addition to the structure of marine ecosystems. Eating animals is easy. The first plant eaters on land were probably these guys, the Casiids, which were basically herbivores, which had this huge fermentation vat. And Wilkinson actually says that the high temperature of the Mesozoic may have been maintained by the methane of dinosaur herbivores. Okay. Certainly cows now produce methane. And the rumen of the cow, the part of the stomach that makes it a ruminant, the rumen is essentially a large anaerobic fermentation chamber where plant-degrading rumen microbiota ferment otherwise non-digestible plant-based foodstuffs into the volatile fatty acids, acetate, propionate, and butyrate. We've seen these short-chain fatty acids before. They're involved in development. They're involved in the digestion of plant fiber into developmentally active products. Well, this is what we see in the rumen. The cow cannot digest plant material. It needs the symbionts in its rumen to digest the plant material. But here's what's interesting. The rumen is made by the bacteria. Newborn calves have sterile rumens. There's a little flap. About 25% of the stomach is rumen. However, within a day or two of birth, the rumen gets colonized by those microbes it got from its mother. And these microbes stay in this non-functional rumen. The rumen does not work as long as the cow is getting milk from its mother. The milk gets shunted down this tube. But once the calf is weaned and starts eating grain, a change happens. The rumen starts to grow because the bacteria that are in the rudimentary rumen digest the plant stuffs into butyric acid, propionic acid, and acetate. It makes the short-chain fatty acids. The short-chain fatty acids cause the rumen to develop. It causes the musculature to develop. It causes the growth of the rumen. It causes the expansion of the rumen. And what Sander showed in 1959 was to make the rumen grow, all you had to do was to give the cow butyric acid. It will induce the growth and the differentiation of the rumen. We now know that butyrate infusion induces the differentiation and morphogenesis of the rumen by upregulating certain growth transcription factors. The transcription factors are upregulated in response to butyric acid. And these may repress repressors of development. They are involved in G protein stimulated growth. They're involved in changing transcription factors. So the rumen bacteria present in the rudimentary rumen since day 2 ferment dry feed into butyric acid. Butyric acid causes the developmental changes that generate the mature rumen. So the bacteria are constructing their niche. This is not only developmental symbiosis, it's niche construction. They are making the rumen. Butyric acid, when given to unweaned calves in their diet or injected into the rumen, will cause these changes. Butyric acid is a nucleosome modifying agent. The bacterial community can now work as nutritional symbionts to convert the grass and grain into simple carbohydrates. You have both developmental symbiosis and nutritional symbiosis. The developmental symbionts have made a new type of organism. They have made a ruminant. So developmental symbiosis is necessary for herbivory. The establishment of symbiosis between potential herbivores and various microorganisms of the gut may have been a necessary precondition for the evolution of feeding on such plant tissues. There are cases where the symbionts are not needed for an animal to digest plants, and these are found in the plant-eating beetles. And here what one has is the horizontal gene transfer of genes capable of digesting cellulose from bacteria and yeast into the plants. These were originally microorganism genes. They are now genes of the beetle, and it allows them to eat the plants. Microbes may have scaffolded major evolutionary transitions in different ways. The origins of multicellularity might have come about by bacterial induction. We know that certain choanoflagellates can become multicellular in the presence of certain bacteria. Similarly, those same choanoflagellates, if you add V. fischeri bacteria, the V. fischeri bacteria make a protein which induces meiosis and mating in the choanoflagellates. So it's possible that the origins of meiosis and sexuality and the origins of multicellularity came about through symbioses. The origins of neurons may have come about through symbioses, because you find that you have the neurons controlling the symbionts. The neurons of Hydra are acting like the immune system, but the Hydra doesn't have an immune system. The mesoderm hasn't even formed yet in Hydra, but the neurons are controlling the placement of symbionts in Hydra. They are acting like an immune system. So the origins of neurons may not have only had to do with feeding and with host defense. They might have to do with symbiont and the roles of symbionts in development. We certainly see the acquisition of viral genomes in the mammalian placenta, and the notion of the placenta being a product of not only the host animal, but also endogenous retroviruses. So we have here a whole new way of looking at biology. It's an imperative for zoology. You can't talk about animal physiology or development without talking about microbes. So the whole genome, the genome of both the symbiont and the host, is important developmentally, physiologically, immunologically, probably behaviorally. Is the whole genome important evolutionarily? Might nature be selecting the holobiont? Might nature be selecting teams? I think the answer will be yes. The genome of both microbes and host is critical in development. Symbionts can provide developmental signals. The genome of the holobionts can be transmitted, and alleles of symbionts can be selected. Niche construction can be seen in the interactions between symbionts and host, and even genetic assimilation can be inferred from lateral gene transfer of genes for cellulose digesting enzymes in the vertical maternal transmission of obligate symbionts. Developmental symbiosis can generate novel evolutionary trajectories, such as the ruminants, and other transitions may have had similar origins in developmental symbiosis. We have to look at the biosphere. We have to look at the critical zone. We have to look at Gaia in terms of symbiosis. We see that symbiosis is the evolutionary strategy that supports life on Earth. You have the rhizobacteria and the legumes with their nitrogen fixation, the mycorrhizal interactions with plant roots and seeds so that they are stabilized in the earth, the endophytic fungal protection against desiccation, coral reefs and tidal seagrass ecosystems sustain oceanic biodiversity. Within these big symbioses are the smallest symbiotic web that we call organisms. And these are the products, I think, of ancient symbioses that we call cells, and of the products of further ancient symbioses that we call genomes. Paper just came out in Cell today. Don't forget the viruses. Endogenous retrovirus promote homeostatic and inflammatory responses to the microbiota. Our skin may serve as immunoprotection against bacteria through its endogenous retroviruses. This is something that remains to be looked at. Most symbiotic studies look at bacteria and fungi. The viruses can't be forgotten. And so I think that developmental symbiosis will be very important as an evolutionary strategy. And so this talk was being given to you by Team Scott Gilbert. And I wish I had one of those, rally jackets that the car drivers had, which show all their sponsors and whatnot, because I am sponsored by Bacteroides, I'm sponsored by E. coli, I'm sponsored by Firmicutes. There are lots of bacteria that are going into this talk today. So thank you very much.
Right. Thank you so much. I think that talk really sheds a light on aspects of, I think, development that very few of us think about because we're so stuck in narrow problems. Thank you so much for that talk, Dr. Gilbert. And so now we could move on to the question and answer session that Sukriti, Nilanjana, Sukanya, and Ayanti will handle. So it's over to you guys.
Yeah, thank you, Dhananjay. Professor Gilbert, a riveting talk indeed. So the first question, there are a lot of interesting questions here. The first one that we would like to pose to you is is from an anonymous attendee who's asking, my question is with reference to mice born in mothers raised in germ-free environments, which showed autism-like behavior. Could it be probable that the rise in diagnosis of autism spectrum-like disorders, especially in the West, may somehow be related to hyper-hygienic environments that the children are raised into?
Yeah, good question. It's really interesting because we're all kind of raised in these hyper-hygienic environments, and people like Martin Blaser have really been saying that we may be losing the symbionts that are necessary for our normal development after birth, such as cognitive developments. And this is being looked at, and certainly there's been a case study, and I don't know if I don't think I have it on my slides. There was a case study that came out 2 years ago in humans that showed that the microbiomes of humans with autism, or at least some of the people with autism, were different than the microbiomes of your people with normal behaviors. And this is important, these people who had severe autism could get their autism levels ameliorated. They could get more functional if they got fecal transplants, stool samples inserted into them from people with normal behaviors. So this is, that was a pilot study, but the pilot study was showing something very fascinating, which was that for a certain population of people with autism, if you change their microbes, their behaviors got more normal. So yes, I think that there is a possibility that we are born into an environment that might not have the microbes that are necessary for our normal development. This has been shown by a group of Finnish researchers who were looking at asthma and allergy. And they found that certain microbes which were in the environment of the farm, our traditional natural environment, the microbes that were in the environment of the farm are different than the ones that we see in the city, and that those ones that are in the farm can lessen our atopic allergies and asthma. So yes, they actually are looking at ways of inducing people to have farmyard-like bacteria in their homes.
Thank you for that answer. Another interesting question that's come up is regarding the colonization of Vibrio that gives the light organ in aquatic animals. The question regards as to how is it that the Vibrio bacteria are able to find squid just after its birth, and is there some sort of a chemical trace that is universal that is able to attract these bacteria to the squid?
Great question. And it looks like there is. That chemical might be chitobiose. The squid is releasing small amounts of kind of a chitin-like compound, a small chitin precursor, into the environment. And that seems to attract a lot of gram negatives. And then once the gram negatives are there, the ones that don't work are kind of winnowed away. And just a few, maybe 8 or so, of the Vibrio fischeri bacteria get in to this little hole that they make, and then continue to make the hole and colonize it. And this is done, at birth, but it's also done every day because the squid gets rid of a lot of its bacteria and then reincorporates some. So there looks like there's both chemotactic and haptotactic factors that are involved in keeping and selecting the bacteria. A lot of the bacteria are oxidized to death. A lot of them are sloughed off by the mucus. So there's a lot going on. And there's been some recent review articles. In fact, there's one in this year's current topics in developmental biology, which go into how the squid gets the bacteria it needs. But it's the bacteria are in the ocean water. They're in the bay water. So the squid doesn't actually give them the bacteria. They're already in the environment.
Thank you for that answer. My colleague Nilanjana will pose some interesting questions to you, I guess. Nilanjana?
Yeah, hello. Hi, Professor Gilbert. It was a beautiful talk. And so I would like to relay you some questions. So we have one question from one of our viewers on YouTube who asked that how different are the species of gut microbiota across different individuals, and can disease associations be made due to the presence or absence of specific species of bacteria?
Oh yeah, that's a wonderful question. And there are like 3 papers that came out this month on that question. There seems to be a core microbiome. And the core microbiome is interesting because it isn't so much a core microbiome based on species as it is on functions. So the notion is that it's the song, not the singer, as Ford Doolittle said, that many species can be used, and it's how they play with each other. It's how they interact with each other. But there does seem to be a core microbiome. And one of the things that's interesting is that the core microbiome seems to be the same on a genera level, not necessarily on a species level. And the core microbiome differs between people, but the species differs between people, and it changes with each meal. That when you eat certain foods, fatty foods, for instance, it will say to a certain set of bacteria, okay, these are the ones that you know how to metabolize, be fruitful, multiply. Whereas if you eat a low-fat, high-fiber diet, a different set of microbes are induced. And so the microbes that you have are ones which depend on what you eat they also depend on your host genome, because the genome that you have will be producing nutrients for the microbes, and certain microbes will be able to use those nutrients better than others. And you might be producing nutrients slightly differently than your neighbor will be producing certain nutrients. And so there's this balance going on between the host and the symbionts. And that could be very important in Helicobacter, where Helicobacter is probably good. Certain strains of Helicobacter are good for some people and bad for other people. You have differences in genome, differences in diet, which both are playing a role. Now, are there differences in diseases that you get with different microbes? That has been much harder. There is a lot of research going on to look at things like Lactobacillus reuteri, which is a bacillus which may be very important in bone development. It might be very important in sustaining bone mass as one ages. There are people who are looking at a certain bacteria which seems to be very important in augmenting metabolism, such that people who have this bacteria may be thinner than other people. And that might be a good in some situations and very bad in other situations. But hey, if one could find a bacteria which in everybody would make you thinner, you can imagine this being a commercial product. So there are many companies now who are looking to find the bacteria which across the board will be good for you. The trouble is different people have different genomes, which will make different bacteria work in them and not in others. I think that the notion of precision genetics is very stupid without looking at the bacteria. Because as I said, we get 8 million different genes from our bacteria, and the bacteria are responsible in large part for our drug metabolism. How we metabolize anti-cancer drugs, how we metabolize anti-epilepsy drugs, how we metabolize anti-heart attack drugs, it's the bacteria which are involved in this. Now, our diet and our genome may predispose us to have certain bacteria, but we differ from person to person to person depending on what bacteria we have. So the notion of looking at our genomes and saying, oh, this is, we'll do predictive medicine for you, I think is a very unsubstantiated idea, because it's the bacteria which are going to become involved big time.
Yeah, that is very interesting to know about. Relating to this question, there's another question that how does some microbes gain the immune privilege, and why doesn't our immune system fight them?
Yeah. There are a number of hypotheses about this, and one of them put forth by Robert Root Bernstein, I think, is very interesting, and that's that the bacteria can either make or hide themselves. The bacteria that become symbionts basically are able to interact with our T regulatory cells and tell our T regulatory cells that they are self. And so that the T regulatory cells don't, they are not going to, that they will continue to suppress the T helper cells that would normally react against them. And so it seems that there may be a case for the symbiotic bacteria having evolved with us over billions of years, evolved with us and our ancestors, that they're recognized as self somehow. And they're looking now at parts of the bacterial outer proteins, the bacterial membrane proteins and outer wall proteins, and seeing motifs in them which are indicative of self. And this is an ongoing area of research, but it is absolutely fascinating that some bacteria that we get, and again, we get them before the immune system is fully matured. And that's another important point. We get these bacteria from our mother. Our immune system has not matured yet because it's the bacteria that cause our immune system to mature. So, but the question is, why is it that this immune system, why is it these bacteria which can cause the immune system to mature in the first place?
Yeah. So going back to the human development, what would your comments be about the gut microbiota of cesarean people born in cesarean section?
Yeah. Again, there was just a paper published this month about it, saying that first of all, those children who are born by cesarean section and who are not breastfed have a higher incidence of returning to the hospitals with respiratory and digestive problems during the first 5 years of life. So there is something about this. It's a statistical thing. It's not an obvious thing, but the statistics show that this population is at higher risk. It's also been shown that those people who are born by cesarean section and who are not given breast milk often get colonized by the bacteria of the hospital or the place where they're being born. And this is not good because the hospital has all sorts of bacteria in it that you do not want. It has all sorts of antibiotic resistant microbes. However, it's been found, and this was the paper that was published about a week or so ago, that if you take the vaginal swabs, if you take paper, or if you take tissue and put it in the mother's birth canal, and then you swab the baby with it, face and lips, that the baby's microbes, that those bacteria are really good at proliferating, and those microbes will replace the hospital bacteria. So the good news is that you can get a normal set of bacteria from a C-section baby by just giving it the mother's microbes, on paper, or you just have the mother holding the baby for a long time.
That is really interesting to know. So with that, I would like to hand over the question answer session to Sukriti, and she'll carry on from there. Thank you.
Hello, sir. Great talk. Continuing with the theme of disease and gut microbiota, Yashashwini wants to know whether we would go so far to draw a correlation between the reported decline in mental health along the generations to the change in gut microbiome.
I don't think we can go that far. We've put so much garbage into the environment, which can affect the nervous system, all these PCBs and insecticides, pesticides, heavy metals, all these things that can affect the nervous system. I don't know if you can lay the blame, if there is blame to be laid. I don't think you can say it particularly on the bacteria. I have scientists who are trying, I have friends who tried to explain the current political situation in America this way, or at least until November, but we don't have evidence for that.
Thank you, sir. There's two related questions. One wants to know, how do you think humans who fed on animals changed when they changed their dietary lifestyle? How do you think the microbiome inside allowed humans to do this? And also when we were colonizing the Arctic, where the plant-based diet is really low, so how did the microbiome change then?
Yeah, I don't know the answers to that. Good questions. I do not know the answers to that, nor do I know where the vitamins come from in the Arctic either. I'm sorry, I don't know. Good questions.
Thank you, sir. As a final concluding question, would you like to comment on the social, cultural, the behaviors, different behaviors that can arise in different social, cultural settings based on the differences in gut microbiome?
Whoa. Wow, that's a question for after dinner talks and over beer. Let me give the most extreme hypothesis. The most extreme hypothesis is this, and it comes from that study that I showed about autism in mice. Maybe it's the fact that humans are asocial animals, that our genome is asocial. But the bacteria want us to survive. The bacteria want us to make more people. The bacteria want us to mate. It's the bacteria that are inducing our sexual fantasies. The bacteria want us to get together because that's how we make more bacterial niches. That everything is controlled by the bacteria. That our social lives are programmed and that we are just the bacteria's way of making more bacteria. That's the most extreme. However, there are all sorts of social situations where bacteria are transferred from person to person. Hugging, kissing will give, a good kiss gives about 8 million bacteria transferred from one person to another. Certainly, if you look at the cultures of food, cheese, yogurt, wine, all are microbial based. I mean, bread and wine, beer, all of them are microbes. And so that the microbes have really helped human culture in fascinating ways. Whether there are differences between human cultures based on microbes might be a product of food differences, in that different cultures, and it's been said that certain people can digest milk better, and therefore they have a dairy culture because of, microbial interactions. I do not know whether this is so or not, but I certainly think that so much, and this is going back to the question that, or the comment that, Vidya and I were talking about before, this notion that you can't separate human history from natural history. They are merging in things like holobionts, and they're certainly merging in the holobiont properties of our culture, especially our food cultures, which is based to a huge extent on our interactions with the microbes.
Thank you. Sir, do we have more time? There are lots of interesting questions in the chat box.
It's entirely up to Scott. We can go for at least 5-10 minutes if it's convenient to him.
Sure, let's go for another 5-10 minutes, sure.
Okay, so Varsha Singh says that there's a lot of emphasis on gut microbiota, but how much do we know about the impact of other organs, including those in the skin?
Yeah, the gut microbes have been some of the easiest to work with because you can get stool samples. But there is a lot of interest in the different medical subspecialties concerning their microbes. For instance, obstetricians and gynecologists are very much interested in the microbes in the female reproductive tract, especially why some people get yeast infections, some women get yeast infections, other people don't get yeast infections. Are there microbes that are protecting the reproductive system against yeast infections? People are looking at microbes that may be in the uterus. And are these microbes in the uterus protecting the fetus against inflammatory responses? There are all sorts of people who are really interested in the microbes of the reproductive tract. People, of course, are interested in the microbes of the skin and how the skin is used as our, protection against the environment. How does skin heal? And are microbes involved in skin healing? There may be very important microbial components to skin healing. And of course, another group of people who are just fascinated with the notions of holobiont communities are the ophthalmologists and dentists, because here we have areas of our body which are really important to the function of our body, constantly being exposed to new microbes. How are they being protected? And are there microbes that we have that are protecting them? And the answer seems to be yes. We do have microbes whose function is to protect the eyes and the oral mucosa. So yeah, we do a lot on the microbes of the gut because that's where you have the highest concentration and the largest densities and the most species. But we do have microbes on every orifice, on every part of the body that is exposed to the outside environment. And there's now a debate on whether there are not microbes in the amniotic fluid. That is going to be, there's a lot of rather heated debate on whether or not we develop in a sterile environment or not. There may be microbes already in the amniotic fluid changing our physiology. That remains to be seen.
That question was there in the chat box. But shifting gears a little to viruses, someone has asked, do bacteriophages play a role in maintaining balance of populations of different species of bacteria in our gut, skin, vagina, urethra, mouth, etc.?
Yeah, this is, the viruses have been ignored for so long, but if you want to talk about sheer numbers, there might be 100-fold more viruses in our gut than bacteria. And these viruses probably do play a role, the phages probably do play a role in determining to some degree the population dynamics of the gut bacteria. In addition, we have these endogenous retroviruses, viruses that we picked up in our evolutionary history that may be responsible for the homeostatic and maybe healing properties of our skin, but they seem to be absolutely critical in forming a new type of cell in the mammalian body. And the mammalian body has a cell in the uterus, which is different from the uterine cells in any other mammal. And that's the uterine decidual cell. And the uterine decidual cell is responsive to progesterone. It's the cell that expands in the uterus to catch the embryo. It's the cell which is involved in the original nurturing of the embryo. It's the cell which makes the uterus capable of sustaining the embryo and the fetus for 9 months. This cell is responsive to progesterone. It may have become responsive to progesterone by progesterone responsive enhancers being brought to certain genes by endogenous retroviruses. If you look at the signature near the progesterone receptor elements of certain genes, you find various you find retroviral signals there. Also, certain proteins which are involved in pregnancy in the trophoblast, the proteins that make the syncytial trophoblast multinuclear. These are viral fusion proteins. These came from viruses, and they're being utilized to make something new, namely the trophoblast. So mammalian reproduction may be predicated on the history, the evolutionary history of viruses. I mean, yeah, we are not alone. I mean, the notion that we develop with the other and that we develop evolutionarily as a cohesive amalgam of otherness, that others have been incorporated into what we call ourselves, I think is just a new way of looking at all nature, an imperative for zoology. Yeah.
Right. And I think with that, that would be the last question from the audience. I was just wondering if Dr. Nanjunde had any questions, or Vijay, if you have had any questions.
Nothing for the moment, thank you.
Right.
There are a million questions, but not for now.
I think...
Thanks for a lovely talk, Scott.
Thank you. And I think that this field opens up so many more questions that it answers right now. This is still a very young field, and it's a field that's just beginning to get its paradigms. It's something which, the people who've been working on developmental biology for years, this is something new to them. The people working on evolutionary biology for many years, this is new to them. I think that this is an area which is potentially incredibly important and which is just finding itself. So, you know, what Symbionts said, you know, about science, that, you know, an area of the best area of science are those areas where we have new technologies that can answer questions that we just are recently discovering. Yeah, I think that's where we are right now. It was where embryology was in 1900. This field of symbiosis and development is now.
I think, I mean, this, like you said, this has been truly fascinating. We have a barrage of questions that we will save for the Slack channel and maybe address and discuss later. And I just would really, really like to thank you for participating in the discussion and giving us this talk. Vijay and Vidya, this was really, really nice of you to join in. Yes. And to all the participants, the audience, we touched about 400 participants at one point. So that was amazing. Thank you very much. We really want to take this forward. I think that we can continue to do some fantastic intellectual exchange in this club. And this was the first in the seminar series. We hope, we want every single activity that follows to be this rewarding and this enlightening. Thank you very much. I would like to thank also Raj and Sonia and oh God, Sachin for actually for being part of this team, for actually initiating this and making sure that this goes forward. So thank you very much. Thank you very much, and hope to see you.
You'll have to buy him a beer now, Dhananjay.
I will. I promise.
Yes,
This is embarrassing.
And best wishes for your new society. Really,
very
I
much.
think it's such a great idea.
Thank you.
Thanks, Scott.
Everyone who's interested in being part of this, please join in for the organizational meeting next week. The details will be on Slack and must have been shared here as well. Thank you very much.
Thank you.
Thank you.
Thank you. Bye. Thank you, Scott. Thank you,
Thanks, Scott.
everyone. Bye.
Bye-bye.
It's been a pleasure to be here. Thank you.
Thank you. Thanks, Scott. Thanks, Vijay. Thanks, Vidya.
Thank you.
Vijay and Vidya.
Thank you.